Okay, so today I'd like to talk to you about a Bioconductor package that I've been working on and a sort of workflow that I'm hoping will be useful for a few different types of data. So this package is called Weitrix. And it's all about a particular form of data, and that's where you have some matrix of observations, and that matrix has an accompanying matrix of precision weights. So this might be familiar from using Vroom in Limma. It's very much the same concept, seeking to generalize it. And this definition of weights, like, it's compatible with Limma. It's also compatible with the LM function in R. So, but this package is specifically about weights for a matrix of data, about choosing good weights, and then once you have good weights, doing some useful things with data of this type. And I've tried to use sort of modern things in Bioconductor like delayed array and parallel. So it can work reasonably well with large data sets, including single cell. Okay, so just to start off with, what do I mean by weights? So this is weights in the context of weighted least squares. They're fitting a linear model based on a set of predictors. We're aiming to estimate some coefficients to best match observations. So we've got observed value minus like predicted value squared, and then like the new element here on top of least squares is we weight particular observations. Now, the ideal weights to have in this situation are setting the weights to 1 over the noise within each observation. So that's optimal in a few different senses. It's what's called a blue estimator, so best linear unbiased estimate. And if the noise is Gaussian, it's also what's called a maximum likelihood estimate. Another reason to choose weights this way is that then the errors in the estimates of these coefficients are going to be asymptotically Gaussian, and we're actually able to work out the variance of the errors. And that'll let us perform valid statistical tests and things like that. And then finally, we can use a weight of 0 to indicate missing data. So this can cope with missing data in a fairly nice way. Okay, so just to dive into this concept a little bit more, looking at this and thinking about like minimizing this weighted sum of squares, the minimum isn't actually going to be like the choice of coefficients that minimizes this isn't actually going to be modified if we were to say double all the weights or halve all the weights or something like that. So there can be we can have weights up to some sort of unknown scaling factor, and that's enough to fit the model. And then once you've fitted the model, you can look at the residuals and like estimate what that unknown scaling factor was. So I'm going to call this the dispersion. This is very similar to like estimating residual standard error, residual standard deviation, sorry, just adding weights into the mix. Now, one thing you need to think about when estimating this dispersion, like that's an estimate, it's imprecise. So that leads us to, rather than using Z tests when we do statistical tests, we'll use T tests, or rather than chi-squared tests, we'll use F tests to take into account that uncertainty. And then, so like, I'll maybe talk a little bit about what limma adds on top of this. So this idea of dispersion, that's exactly part of what limma does. It estimates a dispersion for each row of the matrix, each gene usually. And then the sort of limma special source is shrinking all those dispersions per gene towards some sort of common value, and that gains you some statistical power. Yeah, another place where these dispersions come up is in, like, some GLMs have this dispersion parameter. All quasi-likelihood models have this parameter. And so, for example, the EDGE-R quasi-likelihood method involves fitting GLM models to each row in the matrix, but then again estimating this dispersion and doing the exact same thing as limma with squeezing those dispersions towards a common value. Okay, and yeah, my package Weitrix has various functions along those lines, and they'll also do this sort of squeezing thing. Okay, so one final thing about weights. A common case that I'm interested in is where each observation in a matrix is based on some like lower level observation, and each of these higher level observations will be the average of the lower level observations. I'll get to an example to make this concrete in just a second. But if we think about that case, like each lower level observation, like that might have an associated noise level variance. And then when we think about averaging, we can then work out the variance of that average is going to be this. And so fitting this into our weighting scheme, we would naturally set the weights to be the number of these lower level observations. And that's also a fairly nice way to think about weights. It's as if this data point is actually n data points averaged together. Okay. So let's look at an example. So I'm going to be looking at some PAT-seq data. So PAT-seq is a protocol Schroeder Balhaus invented, and it's a 3' focused RNA-seq method. And often we'll have reads that sequence right through the end of the genomic part of the transcript and then into the poly A tail that's been added on. And then if we're very lucky, we might even get the sequence of the adapter at the end. But the interesting thing here is we get a poly A sequence for each read, or for some of the reads at least. And that gives an indication. It's not a perfect indication, but an indication of the tail length of the transcript. So we can investigate biology to do with that. So what I'm going to do, each read belongs to a sample, and we can also align it to the genome and assign it to a gene. So for each gene in each sample, we can average the tail lengths, and that's going to be the data in our matrix. And then, at least as an initial set of weights, we could use the number of reads involved in each of those averages. Okay, so particular data set I want to look at is yeast. these yeast have been exposed to a pheromone that stops them from dividing, and then that pheromone's been washed away and they've started dividing again. And so this is a time series experiment. It actually goes through 2 cycles of cell division, and then there's 2 strains of yeast involved. So I'm first going to look at fitting a linear model where the tail length, so this is our linear model syntax, like saying tail length can be predicted based on the strain of yeast. So based on two sort of influences on it, the strain of the yeast and the time. And slightly annoying thing about this data set is there weren't any replicates. So this is about the most complex model we can sort of fit to it without doing clever stuff. So anyway, there's a linear model. This is your sort of like your design matrix that you use with lemma. So very much the same concept. And we're fitting the model to each row in our matrix, which is going to be a gene. Okay, so now what I want to look at is some residuals. So this is fit all these models, get residuals, and just pull them all together, plot them in the one plot. So this y-axis is the residual, and then the x-axis is the log read count. Remember, so each of these points is an average over read count genes, read count reads, and so as you have more reads, the residuals get smaller, which is sort of what you'd expect, hopefully. Now, okay, so now I should explain these red lines here. The center red line is an average trend line, and then the top and bottom are plus or minus 1 standard deviation. So now we're going to use read counts as weights initially, and like based on that weighted sum of squares, if we plot the square root of the weight times the residual, that should then be uniform. We want this quantity, square root of weight times residual, to be uniform relative to anything else that we might plot it against. That's what we need for these blue estimates. That's what we need to be able to do valid statistical tests. And so you can see with this choice of weights, well, the left-hand side is looking pretty nice, but then as the read count goes up, so remember we're multiplying these by larger and larger numbers, and it's sort of blown out on the right-hand side. So these read counts are, I mean, they're providing useful information, but we can't quite use them directly. Excuse me a moment. Okay. So how can we get some better weights using the information that we have available? So we've got an initial fit. We've got residuals from that. I'm going to put all of that into a big data frame in long form. So each row is going to represent one residual. And then I'm going to fit a model to the squared residuals, and the predictions from that model, one over the predictions will serve as the new weight. So the predictions are will then be the noise level in each observation, and 1 over the predictions will be appropriate as weights. The particular type of model I'm fitting is a gamma GLM, which is, well, it's appropriate if we believe the noise is Gaussian. It shouldn't be too bad otherwise. And using a log link function, the main thing about that is we expect influences on the noise level to be multiplicative. And then, okay, fitting this model, we have quite a lot of flexibility in what we use as predictors. So what I'm going to do is, so the read counts are in this weight variable. What I'm going to do is fit a spline curve through them. This well-knotted spline, it's, so it's like a natural spline if you've ever used them, with just slightly better choice of knot locations. So fitting a spline curve through the read counts, and then we might also suspect there's like a mean variance trend. So fitting a spline through the mean tail length. So that's the initial predictions from the initial model. Okay. So now this is what our calibrated weights look like versus read count. Now that's looking nice. It's not blowing out like our initial weights did. So this style of plot lets us sort of validate that calibration. My process here would be to plot these weighted residuals against anything I think might be important. So we can also look at weighted residuals versus the mean tail length. And again, there was some trend there. We fit a spline through it, and that seems to have taken care of it fairly nicely. Or one thing I've just started doing and getting slightly uncomfortable results from, we can even break those residuals down, like maybe to individual samples or groups or something like that. And so this reveals a few potential issues in this data, that something a little odd going around this time point, and then the last time point there's something quite odd going on. This is possibly like the model is slightly inadequate. We were limited in the model that we could fit to the data. Like maybe these samples were acquired at slightly different timing, which our model can't account for. Or it may be that PAT-seq has some sample-specific biases. So part of the protocol, for example, is cutting through a gel at a very precise way, and if that wobbled a little bit, maybe that would affect the outcome. So, you know, like one of my big takeaways from this talk is these weighted residual plots are really useful, and you know, maybe if you're doing a VOOM analysis of gene expression data, maybe have a look in this sort of plot. It may show up some interesting things. Okay, so this is quite similar to VOOM, but like we've got this formula that we can specify rather than it just fits a curve through the mean variance. And like, for example, that lets you do things like, even if you're looking at gene expression data, you might like look at some of these weighted residual plots and notice there's some, say, a difference between batches or even individual samples, and then you could like refine your variance model formula, maybe to take into account batches, and then that'll take care of it. I think there's a bunch of variations on the VOOM function that do things like that, but with this function, like it's just specified a different formula. Also note this is like this sort of interface where you specify two linear models. It's really similar to a double GLM, which seems to be something Gordon Smythe came up with, I think, before he got into bioinformatics. So it's sort of interesting. Yeah, I feel like I'm sort of completing a square of things Gordon Smythe has worked on. Anyway, so we've gone through this process. We've hopefully got some well-calibrated weights. What can we do with it? I guess the most obvious thing is to do some differential testing. These are compatible with limma, so if you want to use a limma workflow, you can easily convert back and forth to the limma E-list type. So limma is sometimes a little bit approximate with the way it uses weights, and I'm using weights fairly intensively here. So yeah, there's some gotchas with limma, but yeah, the basic idea is there. So another thing that we especially run into with weights, is that, like, if we're to do some differential testing and then say sort results by p-value, the smallest p-values are sort of as much going to be determined by like genes with high weight as genes with large effects. And more data is never going to fix that. So principled solution to that, there's ad hoc solutions like setting a cutoff on your log fold change, but principled solution is this treat test relative to a threshold. So that's then testing not that the difference is 0, but the difference is larger than some threshold. But that's slightly annoying because you actually have to specify that threshold. So building on treat, I've got a method called Topconfigs, also in Bioconductor, and that basically does a bunch of different treat tests at different thresholds, and then the results are ranked, like the genes are ranked by the largest threshold at which they're significant in terms of FDR. And so effectively, this is giving you a confidence bound on effect size, like a confidence bound on differences, and with an FDR multiple testing correction. So what this looks like, we're a little bit limited in what we can test with this linear model, but this is looking at difference between the two strains that's consistent over time, and we get an effect estimate. So this is a difference of tail lengths, like unit is number of A's, and then there's what I call a confect value. That's a confidence bound on that. That turns up some interesting looking genes. So one nice thing, if we've got calibrated weights, we can draw in some error bars on the accuracy of the measurement. So this is plus or minus 1 standard deviation. Okay, but like we are a little bit limited in what we can test in this data set. So what else can we do? So with well-calibrated weights, we can start looking for excess variation larger than we expected. So for each gene, we sort of expect the dispersion to be about 1. We expect the weighted sum of squares to be about like the number of samples minus the number of predictors, including the intercept term. So, residual degrees of freedom. And to make that more concrete, we expect it to be chi-squared distributed with like residual degrees of freedom. So that allows it a test for anything with variation greater than expected, but one of the things I'm trying to get at with this package is not just rejecting a null hypothesis, but actually quantifying things and putting confidence bounds on things. So what I've come up with is we can do a test relative to a threshold with a non-central chi-squared test. And then based on that, plug it into Topconfigs, and that'll give us confidence bounds. So working out the non-centrality parameter for this test, I wanted these tests to be comparable between genes. So thinking about if there's a perturbation not accounted for by the model or by noise with some certain standard deviation, we can think about how much that weighted sum of squares would be increased by. It's approximately this. There's a little bit of hand waving, but I think this is reasonably good. And like the sort of caveat here is this weighted sum of squares, it's going to concentrate on where the weight is highest. So that may influence the results a bit, but I think in general, this should work quite well. And so what this looks like, so what I'm going to do here is look for genes that have excess variability compared to a very simple model with just an intercept term. So compared to a model that says all the tail lengths are the same. And so that'll pick up on any sort of variation over time, in different strains, maybe some weird combination of that, of the two. So you can see it picks up lots of genes changing over time, a few with differences between strains. If you go far enough down, you even see starting to see a bit of these two cell cycles that went on. So the cell cycles are really obvious in terms of gene expression. That doesn't seem to be the main thing going on in terms of tail length, but there's a little bit, and that's interesting. Okay, then, so one final tool, once you have weighted data, is looking for components of variation. So this is sort of the weighted version of PCA. So the idea here is we'll find several components of variation, and then a component consists of a set of what are called scores for each sample, and then a set of loadings for each gene. And then the contribution of a particular component to, in this case, the tail length in a particular gene for a particular sample, you get that by multiplying the score by the loading. And so you can add together the contribution of each component, and then there'll also be an average tail length, and that tries to predict the data as well as possible in terms of weighted sum of squared errors. So I've done this function. It does automatic varimax rotation. It tries to keep the components as sort of distinct as possible, or sort of sparse as possible, and hopefully that means they're interpretable individually. So this is looking at the scores of the three components, sort of plotted in a way that's comparing them to the experimental design. You can see it seems to be a pattern of increase or possibly decrease over time, difference between the strains, and then this third component, when I asked for 3 components, seems to be getting at the cell cycle. And we can also see that funny stuff that was happening at the last time point we saw in the weighted residual plots. And then if we're particularly interested in this component, we could look at top confident loadings, and that picks out an interesting set of genes. So once your data is in this weighted matrix form, you have all these different ways of interrogating it. Okay, so now just to demonstrate that this is useful for something other than poly-A tail length, I wanted to talk briefly about alternative polyadenylation. So we could do this with PAT-seq, but mixing things up a bit, I want to look at a single cell data set. This is 10x data. 10x, turns out it's another of these 3' focused protocols. Excuse me. And in particular, this is a spermatogenesis data set. It has this lovely T-SNE layout. And so like the development is like clockwise around this. And then if we look at depth of coverage of reads, like over this development course, for some reads we see, say, a switch from a longer transcript to a shorter transcript. So the actual cleavage and adenylation site is probably like sort of a little bit to the right of these peaks. But you can clearly see there's a switch from one site to another. And like we see this sort of APA to do with various things to do with sort of developmental stage, or whether a cell is dividing quickly or not, that sort of thing. So there's starting to be a few pipelines that investigate this. There's SCAPA, there's Sierra, and there's one we've been working on. So David Powell did sort of initial work on this. Sarah developed it into like the Python part of this pipeline, and I've been working on the R part of this pipeline. And we're doing something a little bit different. So David found there were actually reads that have like non-templated poly-A sequence. So we can actually find those reads and like call the APA sites like read level resolution based on reads that have non-templated A's. But then we do like UMI counting at the called peaks. Okay, so now I wanted to quantify APA for each gene. And one thing I wanted with this was a signed measurement, because like we see, and this is maybe a little bit different from, say, looking for differential isoforms. We see like there's these general like global shortenings or global lengthenings. I wanted that to be sort of clear when we look at this. So the way I'm going to do this, and there's various justifications for this, but I'll describe it first. So each gene is going to have a collection of sites from upstrand to downstrand. What I'm going to do is assign each site a score between -1 and 1. And I'm going to do it in this particular way. So the probability that another randomly chosen site is upstrand of this site minus the probability that a randomly chosen site is downstrand of this site. So like upstrand sites will generally get a negative score, downstrand will have positive scores. If you were to sort of average this over all reads, the average would be 0. So it's a little bit complicated, but it lets us cope with, like, if we get a funny situation with a bunch of sites, we can get some sort of quantification of that. If we limited ourselves to looking at 2 sites, we could come up with various other things. But in any case, we've got this scoring scheme, and then each read, sorry, each UMI belongs to a particular site in a gene. So for each gene in each cell, we can average the score for each UMI. So anyway, that's the way I've chosen to quantify it. And now we want to be able to work with this sort of data. One thing we can note about it, we have some idea of the per UMI variance. If we were to assume it was randomly chosen, we could work out the variance. Like if there's a big shift, then this will be a bit of an overestimate, but it's sort of the best I can do. And then of course, we also have the UMI count. So again, I'm going to provide this information to the calibration, per read variance, and also read count, and we can like allow for biological variation by fitting a spline through it rather than just using the read count directly. And again, plot lots of residuals. Few points I want to make with this. I apologize for just showing a bunch of graphs here, but my main point is always be using these weighted residuals. I think they're a very useful tool. Another thing is, like, the residuals don't need to be Gaussian. So for example, like here, these residuals, there's a lot close to 0 and then a few that are large. That's fine so long as this standard deviation is uniform. And we have plenty of observations here, so like, there's plenty of chance for central limit sort of thing to kick in. One thing we do need, and that I was very careful picking this particular way of quantifying APA, these observations need to be unbiased. And in particular, we don't want like a low precision observation to be a bit biased and then a high precision one to be unbiased. It would be tempting if you've got a low precision estimate, maybe we could squeeze it towards the mean, but that's something we don't want to do. That it's much more important that the observations are unbiased and that they're minimum error, because that's what we're relying on in order to like do statistics with this. Okay, so and then just saw this already with tail lengths, but you can do all the sort of stuff we were doing with tail lengths. Finds a bunch of interesting genes. I guess the main point I want to make here, like the top gene by effect size or confident effect size here, there's actually a lot of missing data, lots of cells where we just didn't see that particular gene. But that's fine. It's like in the cells we did see an effect, it's quite large. If we were to look at the most significant, like in terms of rejecting that the APA is exactly 0, we'd look at this gene instead. It's this one down here. But that would just be on the basis of there's a lot less missing data, and therefore we like the standard error of the estimate is is much smaller. Okay. So, that's the package. Hoping it's going to be useful for different data types. Hoping like it's sort of with these sort of differential testing, like you can go very theoretical with your nice negative binomials, or you can go sort of more pragmatic. This is towards the more pragmatic end of the spectrum, but with quite a lot of flexibility, because we can like specify this we can come up with weights based on a linear model formula. I think these weighted residual plots are very useful. Even if you're using some fancy statistical notion, it's good to actually plot these residuals and make sure that matches reality. And then once you have your data in this form, you can do various useful things. And yeah, I guess another general theme here is like it's a workflow based around measurement but confidence bounds. You don't need p-values to appear in your outputs anymore, but still keeping the same like rigorous statistical theory underlying it all. Okay, so I'd like to thank, Gerda Beilhaas came up with the PATSEQ protocol, has produced large amounts of data. I've been working with her for quite a few years now. Bernard Dichtel was involved in the generation of the particular PATSEQ we looked at. Sarah Williams developed a large part of the single cell pipeline, which we were looking at results from, and David Powell was also involved in the initial work for that. So, thank you.